The overall goal of the protocol is the production and purification of human norovirus protruding domain in E. coli in order to obtain extremely pure protein for X-ray crystallography.
Our protocol for protein expression, purification and crystallization is fast, robust, and can be used for structural and non-structural proteins. The main advantage of this protocol is that our proteins are very pure and can form crystals that can diffract to high resolution.
According to our experience, the protocol might need adjustment for less soluble proteins. For example, the growth medium or growth temperature during protein expression can be changed.
To begin, transform 1 microliter of the P-MAL-C2X vector coding for the MBP-HISP domain fusion protein into 50 microliters of competent E. coli BL21 cells using a standard transformation protocol. Add 600 microliters of SOC medium to the cells and grow for 1 hour at 37 degrees Celsius. Subculture the transformed cells into 120 milliliters of Lb ampicillin overnight at 160 rpm and 37 degrees Celsius. Inoculate 9 liters of Lb ampicillin with the subculture. Grow the cells in the incubator at 37 degrees Celsius with a shaking speed of 160 rpm until the optical density at 600 nanometers reaches 0.4 to 0.6. Subsequently, lower the temperature to 22 degrees Celsius for approximately 1 hour.
It is important to wait until the temperature reaches 22 degrees Celsius before inducing with a low amount of IPTG.